Assalamualaikum and greetings to judges, lecturers and all participants. Today I'm going to present about my finding on my research entitled Early Ontogeny and Embryonic Development of Silver Barb Barbonymus gonionotus. My name is Anissa Ainnina. This study was supervised under Dr. Nadiatul Hafiza from Department of Biology, Faculty of Science, UPM. My study focused on the embryonic development that are divided into early development, which is from cleavage to gastrula, and late development from segmentation to hatching. These are observed within certain interval that are vary between species. In this study, we choose Barbonymus gonionotus, locally known as lampam Jawa, due to their importance in aquaculture industry across Southeast Asia. Due to their popularity, this species has been widely used in aquaculture, especially in Malaysia. Although several studies has been done to examine their advanced biology like nutritional requirement, reproductive biology, however, information on their basic biology are still lacking. This information including the information on the early embryonic development, which are very crucial for their breeding and also early culture management. The objective of this study is to describe the stages of an early embryonic development of silver barb, which is from fertilized egg to larvae, and at the same time to assess the timing of important developmental stage from fertilization to larval age in Barbonymus gonionotus under laboratory condition. This experiment started from the broodstock selection, where they are transferred from the wild habitat to spawning tank in Department of Aquaculture, Fakulti Pertanian, UPM. The spawning tank were equipped with gentle aeration, and the fish were allowed to acclimatize for about 8 hours for them to adapt to their new environment. The female fish will differ from the male fish by having a slightly bulging bellies. And then, the female fish was injected with 0.5 ml per kilogram artificial ovotide hormone to induce maturation of egg, while the sperm from the male broodstock were gently stripped. And then the sperm and the egg were mixed together in a bowl, and it mixed using a feather. And then after that, they were transferred into 5 replicated tanks equipped with mild aeration. The water temperature in the tank were maintained between 23 degrees Celsius to 26 degrees Celsius with pH between 6 to 7. The fertilizing egg were taken from the breeding tank using a plastic dropper to avoid any pressure that can rupture the egg membrane. And then the fertilized egg were placed in the clean petri dish containing water to be examined under compound microscope Leica DM570 fitted with a pre-calibrated micrometer eyepiece supported by dyno eye. For result and discussion, this is a table of embryogenesis of Barbonymus gonionotus under laboratory condition. Early embryonic development started from early cleavage stages from 1 cell to 32 cells. 10 minutes after fertilization, 1 cell stage was observed, with a germinal disc begins to form. And then 15 minutes after fertilization, 2 cell stage was observed, with a 2 equal half blastomere called as meroblastic cleavage. And then 34 minutes after fertilization, egg cell stage was observed with 2 rows of 4 cells. And then at 15 minutes after fertilization, 5th cleavage produced a globule-shaped blastomeres. After the early cleavage stage, morula stage begin. At 58 minutes after fertilization, early morula stage was observed. With irregular cleavage, it will produce rounded ball of cells. During this stage, the blastomere were extremely hard to count, and it only appeared as a rounded ball of cell. At 1 hour 30 minutes after fertilization, late morula was observed. The blastodon will consist of outer and inner layer, but there are no major change compared to the early morula stage earlier. And at 1 hour 25 minutes, early blastula was observed with a bulging blastodon. As you guys can see here, this is the blastodon. And then 2 hours 23 minutes after fertilization, the late blastula was observed with the blastodon extend slightly over the yolk. After the blastula stage, gastrula stage begin. At 3 hours 8 minutes after fertilization, early gastrula was observed. The cell at the vegetal pole start to invaginate into area. So this is the vegetal pole, and then this is the animal pole. And then at 4 hours 20 minutes after fertilization, late gastrula was observed, where half of the yolk covered by blastodon as epiblast. And then at 5 hours 80 minutes, bud stage was observed. During this stage, the head and tail were appeared. Next is segmentation stage. So during this stage, the body start to be segmented. So it consists of 6 somite, 11 somite, 25 somite, and 33 somite. This stage took a little bit longer than the other stage. Like for 6 somite, it took about 154 minutes, and 93 minutes, 84 minutes, 160 minutes, compared to the early cleavage stage, which only take about 10 to 15 minutes each. The last stage in early embryonic development is hatching stage, where it appears with transparent larvae with large eyes, and this was observed 15 hours 5 minutes after fertilization. So basically, the early embryonic in fish start with early cleavage stage from 1 to 32 cells. In fish egg, the cleavage only occur in blastoids. In blastoids, there is cytoplasm that will become embryo, and this type of meroblastic cleavage we call as discoidal. An early cleavage division, it have a highly reproducible pattern of meridional and equatorial cleavage. Meridional cleavage is when it cut the blastomere into 2 equal halves, and this stage has a rapid division that takes about 10 to 50 minutes each. Like from 1 cells to form 2 cells, it only take about 10 minutes, and from 2 cells to form egg cells, it's only take about 15 minutes, and from egg cell to 32 cells, it takes only about 9 minutes. So after that is morula stage. Morula stage observed when the fifth cleavage produced the embryo with many blastomere due to the irregular cleavage. And morula stage was defined by a successive cell 28 division, which is more than 64 blastomere, and it organized in a half-berry-like form. Next is blastula stage. So it was characterized when the periblast and blastoderm regions become clear, and the animal pole was in a cup-like shape. This stage was observed at 128 cell stage, and it changed shape from spherical to oblong, and the yolk sensor layer was formed when the closest cells start to fuse. Next is gastrula stage, where it happened at 15% epiboly. Within 50 minutes of 50% epiboly occurs, the thickened marginal region will form a germ ring, where later it will develop into ectoderm, mesoderm, and endoderm. At a slightly later period, the embryonic shell is formed. So in early gastrula stage, this is the vegetal pole and animal pole. And then at late gastrula stage, half of the yolk is covered by the blastoderm as epiblast. The major morphological change during bud stage is the head and tail region begin to observe. So the embryo was slightly elongated, and it have a roughly bean shape. The tail bud emerged from the posterior neural tube along with the underlying mesoderm. So at somite formation, when the epiboly is complete and we observe already the tail bud, the first somatic furrow is formed. During this stage, we can observe the eye vesicle. And then as the number of somite increase, the linear measurement of the head and also the segmented body part will also increase. And at the same time, the measurement of the unsegmented body part will decrease. During this stage, the embryo will increase in size, where the 6 somite were observed during the middle period of it, and it will continue to 11, 25, and 33 somite. And during this stage, the head and tail end were distinct compared to the bud stage earlier. So this is the eye vesicle. Yeah, this is the head. So we can actually see the head and also the tail compared to the bud stage image earlier. And then we reach to the last stage of early embryonic development, which is the hatching stage. During this stage, the larva appear to be transparent and possess a large eyes. The larvae become more active for them to hatch. And during this period, the yolk sac will completely disappear, and the swim bladder was then filled with gas that enabled them to swim. So in conclusion, the embryonic development stage of Barbonymus gonionotus can divide into 8 stages, started from early cleavage, morula stage, blastula stage, gastrula stage, bud stage, segmentation stage, hatching stage, and larvae stage. Each of the stages were described based on the noticeable or major structural change. And the embryonic development of Barbonymus gonionotus took from fertilization to hatching took about 15 hours. And for future studies, in this study, we do not measure the egg in every stage and also the standard length for the eggs and larvae. So for future study, the comprehensive embryonic development study can be fully conducted by include the measurement for the egg in every stage and the standard length for the eggs and larvae. This is the reference that guide me during my study and also during this presentation. So thank you so much for lending me your ear, and that's all from me. Thank you.